Hello, my name is Marie Gomez and my partner is Jenna Morales, and we do research under Dr. Francisca Sandmeyer. And our research is comparative leukocyte counts among different tortoise species and upper respiratory tract disease. To further understand our research topics, I thought it was adequate to state our three different research topics, which is tortoise differences, URTD, which is upper respiratory tract disease, and white blood cells, which we will interchangeably use leukocytes. There are 5 different species of tortoises in North America, and 4 of which are found in the United States. The Mojave and Sonoran desert tortoises have limited water supplies since they do live in deserts. The gopher tortoises, which live in Florida, are much more exposed to water and nutrient-rich vegetation year-round. The Texan tortoises that live in various climates live in shrublands with slightly greater changes in the forage availability across the year. A slight introduction to URTD, which is upper respiratory tract disease, is that scientists believe that it may be indigenous and flare up when stressed. This is due to several studies done by Ayeyo and several others that transmission rates of 0.5 probability took 45.9 hours of contact to be able to obtain URTD. It is also due to different densities of populations and different habitats that may have an impact. So some tortoises may not even have those 45.9 hours of contact to be able to obtain URTD. So this is why researchers believe that URTD is indigenous and flare up with stress. Some of the physical signs of the nasals include inflammation, softening of cells, or soil-filled nasals, such as the picture featured on the slide. The red blood cell portion of our study was very interesting as we did the counts manually. We got to learn what each white blood cell did. So the heterophils and eosinophils, their main function is primary quick inflammatory response. The basophils take a part in allergic response, and they protect mucosal surfaces such as the nasals. Monocytes, their main role is macrophagocytic activity. Lymphocytes kill viruses and secrete antibodies, and thrombocytes help clot blood and create platelets. So the top left picture in the slide is a heterophil, and right next to it, on the top right, are a pack of eosinophils. They do like to clump together, is what we saw. The bottom right is a lymphocyte, and then the bottom left picture on this slide has actually several of our white blood cells. Our hypothesis of focus was that we hypothesized that there would be differences in white blood cell counts among all the 4 species studied. And why was that important? It is because this study can lead to future studies to find further information on how the immune cells of tortoises operate when responding to URTD. It is also important to know that lymphocytes are known to reduce URTD.
For our methods, we quantified white blood cells from previously collected samples. White blood cells were identified as heterophils, basophils, lymphocytes, monocytes, thrombocytes, or eosinophils based on their morphology. Excluding the thrombocytes, we took our data and did different statistical analyses. We first used ANOVA to determine the differences among the 4 species. Tukey's pairwise comparisons were then used when we saw significance to determine specifically which species differed from the others. We also used principal component analysis to look at the correlation and relationship among the white blood cell types. The top picture on the right shows an eosinophil, and the bottom picture, the cell that looks very similar, is actually a heterophil. Here are the results for heterophils among the 4 different species. We did see significance. Our statistical analyses showed that the gopher tortoises differed significantly from all other tortoise species, the Mojave, Sonoran, and Texan tortoises. Results for basophils were also significant. This showed that the Texan tortoises differed significantly from all other species as well, the Mojave, Sonoran, and gopher tortoises. Our results for lymphocytes were also significant. Mojave tortoises were shown to be significantly different from the Texan and gopher tortoises, while the Sonoran tortoises were significantly different from gopher tortoises. Our monocyte results showed that gopher tortoises differed significantly from Texan and Mojave tortoises, but they didn't significantly differ from Sonoran tortoises. Eosinophil results showed that the Texan tortoises differed significantly from all the other species, and so did the Mojave tortoises. They were significantly different from the Texan tortoises, gopher tortoises, and Sonorans. In our principal component analysis, principal component 1 was determined to be lymphocytes and basophils. Principal component 2 was determined to be heterophils and eosinophils. This shows that principal component 1, the lymphocytes and basophils, had a greater role, consisting of 35.9% of the variation among our data. We also did an ANOVA of our principal components by species, and we did see significance with principal component 1. This showed that the Mojave tortoises differed significantly from all other species, and so did the Sonoran tortoises. They differed from the Mojave as well as the gopher and Texan tortoises. Moving on to our discussion, what we did was quantify white blood cells from 4 different tortoise species. Signs of URTD and the bacterial presence of Mycoplasma acazesii were recorded. We did see significant differences in white blood cell counts between the species. The most interesting result was in the lymphocytes, which showed a correlation to the geographical range of each species. For our other leukocyte types, while we did see significant differences, we didn't identify any obvious patterns that we could relate to their environment. We would like to do further research to investigate URTD and white blood cell activity in tortoises.
We would like to acknowledge CBASE, the biology department at CSU Pueblo, Dr. Elizabeth Peterson, Dr. Chava L. Weitzman, and Dr. C. Richard Tracy for all our efforts towards this study. These are the references that were used to create this presentation, as well as the study.
That concludes our presentation today. Thank you so much for watching, and we would be happy to answer any questions.